Navneet, how many minutes more? Shall we start? I would assume.
We can start. Yeah, now it's 4:05, so we can.
Wonderful.
Yeah.
Okay, so good afternoon, everyone, and welcome to our seminar series of the Cambridge Center for AI in Medicine. I am Mihaela van der Schaar, and I'm the center director. By now, I hope you have already heard about our center, which is aiming to develop novel artificial intelligence and machine learning technologies to revolutionize biomedicine, medicine, and healthcare. And if you want to hear more about us and the activities we are doing and the research that we are developing and get engaged with us, please visit our website. Today, it is my great pleasure to welcome as part of our distinguished seminar series, Dr. Carsten Niemann, who is an associate professor at Copenhagen University. And it is really my great pleasure to really welcome Carsten, because he is a unique blend of clinician that has already applied machine learning widely to a variety of areas, which I'm going to mention next. So he's a board certified clinician and specialist in hematology. And his PhD thesis was also in the area of medicine. But in the past several years, he has really developed a variety of technologies on the basis of machine learning and applied them in diverse areas of clinical research and clinical medicine, both using genetic data as well as more general electronic health record data. He has also used for the first time machine learning to empower clinical trials, and also use them for precision medicine as well as for education. So it is really my great pleasure to have Carsten tell us about his research and his view of how machine learning can be used to empower clinicians, medical researchers, and patients. Thank you, Carsten.
Thank you so much, Mihaela, for that very nice presentation. And Navneet, I think you need to let me share the screen. Thank you. And hopefully you should be able to see my screen now, otherwise let me know. So as Mihaela mentioned, I'm a medical doctor of training and also did my PhD in the lab. During my time at the National Institutes of Health in Bethesda in the US, working under the guidance of Adrian Wisner, also in laboratory and clinical research, I realized the need to better use and organize the data that we generated during research projects, and that's essentially what's initiated my interest to provide what I consider actually pattern recognition across different types of medical data to individualized treatment approaches across different diseases, but mainly focused on chronic lymphocytic leukemia. And what I'm going to present to you today is research, obviously funded by Novo Nordisk Foundation and the Danish Cancer Society. But I want to state also upfront that as a clinician developing clinical trials, also I collaborate with a number of different pharmaceutical companies and obviously a number of different research groups across the world. And I'm actually situated in Brazil today, where I'm working part-time at a hospital here and continuing part-time in Denmark with my research group. So you might consider, why should you find interest in chronic lymphocytic leukemia and CLL? And I believe it's a very good model disease, and I'll try to take you on the journey it has been to develop predictive algorithms in CLL and explain the interaction between data scientists and physicians and laboratory scientists in the development of these algorithms, and here focusing on what we consider CLL-TIM or CLL.
I'm sorry to interrupt you, Carsten, but we are seeing your slides in the presenter's mode. So
I will try to switch then. Thank you for mentioning. No, not at all. So let me see if it gives you the other way around here, or still not. No?
No.
I'll just try to switch off one of the screens then. It's just me having two screens here. So
sorry to interrupt. On the top, there's some buttons that you can select the mode already. So it's not in English, but in the top, there are 3 buttons that probably you can select. Yeah.
So this should allow me to change it. Just one moment, I'll just stop sharing and does it work better now?
Perfect. Fantastic. Thank you.
Great, and thank you for mentioning. So the reason I want to use CLL as a model disease and believe it can actually exemplify a number of different situations in different diseases is that we have a continuum for the pre-malignant state, that's monoclonal B lymphocytosis, which you see all the way to the left. That is a pre-malignant state, actually quite frequent. More than 10% of men and 5% of women above 65 years of age actually have monoclonal B lymphocytosis. And only around 1% of them will develop CLL afterwards. And even the patients developing CLL, chronic lymphocytic leukemia, the majority of those patients will not need treatment at time of diagnosis. They'll be in what we consider the watch and wait position. And over time, approximately half of these patients will eventually need treatment for CLL. So we have this continuum, meaning that we can actually predict prior to an event, because we can actually identify the patients prior, that these patients might be a high risk for a specific event. It could be needing treatment for CLL, or it could be adverse events to treatment, or it could be infections due to the inborn immune dysfunction which comes along with CLL. Thus, the first step in terms of trying to develop an algorithm, a predictive algorithm in CLL or any other disease, would be to identify what should be the outcome. And I've spent days together with Rudy Agius, who was the lead on the machine learning development for CLL-TIM. I've spent days with him discussing how to define a specific outcome that we wanted to model, whether it would be an infection, but should it be based on a diagnosis code from a hospital record, or should it be based on something else? And I'll get back to this, because I think this is really where we need to engage in discussions and communication between clinicians and data scientists and laboratory scientists. But I want to take you a step further back. This is the fraction of patients diagnosed with CLL in different decades or different periods of time since 1978 up until 2013. And you see over time the mortality decreases. But what is more important is if you focus on the yellow-orange part, that's patients dying from infections. Even though the mortality for all other reasons decrease over time for these patients, you'll see that the orange-yellow part actually stays quite stable, indicating that even though we improve significantly the treatment for patients with chronic lymphocytic leukemia and the supportive care, we have not been able to lower the risk of dying from an infection. So this was the background for us to work further in the field of infections in CLL to actually identify a clinical problem that caused mortality for our patients. Furthermore, looking at prescription data, another way of assessing the burden of infections in CLL. And here, starting from time of diagnosis of CLL, that's time point 0 all the way to the right in these 4 graphs, and going backwards prior to infection. What you see in the left blue graph is the prescriptions of any type of antimicrobials for patients diagnosed with CLL prior to the diagnosis, and compared to a matched population, matched on age, gender, and municipality and comorbidity status. So what you see here is actually that up to 7 years prior to diagnosis of CLL, we can detect an increased prescription of antimicrobials, indicating an immune dysfunction and increased risk of infections. And if you look at specific types of antibiotics or antimicrobials to the right, you'll see for some types of antimicrobials, it's actually up to 2 decades prior to diagnosis of CLL. So it's something that appears prior to the diagnosis, this increased risk of infections. Unfortunately, because we actually wanted to define what was coming first, the CLL disease or the immune dysfunction, we could also in a recent paper detect the CLL clone up to 2 decades prior to diagnosis of CLL. Thus, still we don't know what would be the hen and what the egg in terms of immune dysfunction and clonal development in CLL. One approach to lower the decrease of infections in CLL could be to treat patients early on. This has been tried several times, and what I'm showing you here is actually a clinical trial to the left at time of treatment need in CLL. And as you see on the 2 bottom lines here, patients treated for CLL have an increased risk of infections. And if you go for elderly patients above 65 years of age, on the more intensive treatment FCR here, approximately half of the patients would have a serious infection. And if you look to the right here on the graph, this is taking all patients in Denmark from the registry receiving treatment at time 0. And as you see, 50% of these patients would have experienced a serious infection, here defined as having a blood culture drawn, because having a blood culture drawn in Denmark means that you actually had a clinical infection, whether the result from the blood culture would be actually to demonstrate the bacteria or not. And as you see here, 50% would experience that based on real world data. So treatment is not the only choice for these patients, because if you treat the CLL, they would have a high risk of infections. And this even holds true with novel targeted agents, here looking at patients again from real world data from Denmark being treated with ibrutinib. Looking to the left, you see the blue curve representing respiratory tract infections, and within the first year, approximately a third of the patients would actually have had a respiratory tract infections, urinary tract infections, and skin infections below, and yellow and green is a little lower. And what you see to the right, these patients on targeted novel agents actually have quite a high number, approximately a third of the patients within the first 2 years that discontinue treatment. Again, meaning that we have not solved what we can do for these patients. So we should not just start everyone on targeted treatment early on. And a clinical trial still not fully reported from the German CLL study group demonstrates that the frequency of infections during the first 2 years on treatment with targeted agents like here ibrutinib does not lower the risk of infections compared to the control group. So essentially we set out to identify patients at time of diagnosis of CLL. We did not dare to go all the way back to monoclonal B lymphocytosis, the pre-malignant state, because we didn't want to put a lot of people without any knowledge of having a disease, a pre-malignant state, on a clinical trial. And we aimed to develop a clinical trial where we could actually use an algorithm to select patients in need of treatment. But what should be the basis to identify these patients? So essentially, we wanted to start out with whatever data available for these patients. And that's why you see here a photo of my family on skiing vacation. It's been a while since we could go on this due to COVID-19. And it's just to illustrate that we could actually use whatever kind of data that we would have available for patients with CLL. It might be smarter to use a microphotograph of blood film from a patient with CLL to identify patterns that might correlate with a specific outcome, risk of infection, and need of CLL treatment. But we would need a way to also address time series. There's a lot of work from Mihaela's group and another of other research groups has worked with time series. And what you see here is a spaghetti plot for 1,000 patients diagnosed with CLL. They're all aligned in the middle of the photo at the time point of diagnosis. And you'll see before diagnosis to the left and after diagnosis to the right, the development of lymphocytosis, lymphocytes being the main disease part in CLL over time. And the yellow patient is just a random patient chosen to show that these patterns of lymphocytosis over time differs a lot. But no physician today would be able to look at these patterns and identify different groups of patients that might have a specific risk. So essentially what we aimed to was to look at the data available and detect such patterns. In addition to just regular data from medical records and laboratory results, we obviously also wanted to include omics data here, genetics for targeted NGS panel for lymphoid malignancies that we applied across a few hundred patients with CLL. What I want to point out is that most work in terms of machine learning or pattern recognition for omics data would just use these genetic data, whatever type of laboratory data. They would not connect or add these data on top of the clinical data available. And I'll get back to later on the risk of developing quite sophisticated genetic analysis to just identify something that was already apparent from the clinical data in the electronic health record at hand. So again, I want to emphasize that when we develop new omics data, we obviously need to include those on top of what we have already. What you see here to the left is like the visualization of all the different genetic events across this population of patients with CLL. In the middle lower part, you see these genetic lesions being assigned to different signaling pathways in the CLL cell. So trying to use our a priori knowledge to simplify and operationalize the overwhelming data that we have. And to the right, you see that we assigned patients with CLL to different risk groups based on the number, not of driver mutations, but the number of signaling pathways being impaired by driver mutations. And we get a very nice division based on the number of driver pathways being impaired, correlating to time to first treatment for patients with CLL. So obviously we want to include such data in an algorithm. But we also want to include the host factors, not just as has been done in some studies based on genetics and mutations or variants in the germline, but here based on the medical history of the patient. It's here represented by ICD-10 codes, meaning that we had patients either seen in the outpatient clinic or at the hospitals. We didn't have access to the ICD-10 codes from general practice, and prescription data to try to capture the amount of comorbidity that was actually treated at the general practitioners. And what you see here to the right is overall survival for these patients. It makes sense that most comorbidities would actually cause increased risk of dying. But what you see to the left is the interaction between the CLL disease and the comorbidities, as the first bar is representing patients dying from infections. And we know the risk of dying from infections is increased when you have CLL, but in the middle, where you have one comorbidity, it increases further. And if you go to the right, where you see more than two or more comorbidities, it increases further, indicating the interaction between the CLL disease itself and the medical history of the patient. So we also need to include this when we start modeling. So that was the setup for modeling the risk of infection or CLL treatment within 2 years from the diagnosis time point. So again, the importance of the physician, the lab scientist, and the data scientists to discuss and develop the exact definition of the outcome. And because it was based on registry data, I'll come back to later why we did not base it on directly on data from the electronic health records. Because it was based on registry data, we needed to define infection in a way where we could actually operationalize it. And we used the definition as having a blood culture drawn as indicating the decision of the clinical physician that this patient had a clinical infection according to guidelines in Denmark. And then we collected what data we would have available up to 7 years prior to diagnosis of CLL, and we allowed a 3-month period from time of CLL diagnosis because we wanted to make sure that patients that had obvious treatment need would start treatment prior to being assessed by the algorithm, and we want to make sure that all diagnostic workup from time of CLL diagnosis had been finished. And we were able to assemble 1,800 different variables for these more than 4,000 patients diagnosed with CLL from the nationwide registry in Denmark. These data included pathology, diagnosis codes, microbiology findings, all types of routine laboratory results, and the results from regular diagnostic workup of CLL, which would include IDH3 mutational status and genetic assessment for TP53 mutations and FISH abnormalities in terms of cytogenetics. And we used these 1,800 variables to create more than 10,000 different features. Some of the diagnosis codes we modeled as bag of words representations. Then we wanted to model the time distribution of infections and other events diagnosis codes. And obviously we wanted to develop to represent to the model the development over time in terms of different routine laboratory values. And lastly, we wanted to model also the missingness not of random in terms of having a blood test done or a visit to the hospital, because we wanted to include in the modeling the decision by the physician and the patient whether a new laboratory test was needed or not. So this is again, the development of features need to be a discussion or an interaction between the data scientist and the physician, and sometimes also the laboratory scientist. So essentially working on more than 10,000 features generated from more than 1,800 variables and training more than 3,000 algorithms might look like a black box fishing expedition. And that's the usual comment I would get when I present our work to fellow medical doctors, that this is just what everyone warns you against when you start to do epidemiology research, that you just look for whatever correlation you could find in your data. Obviously, to most of you, it will be evident that this is not what you do when you actually model pattern recognition. To make sure that we had a more robust way of assessing the data, we did not go just with one algorithm, but made a genetic algorithm selecting the optimal response across the thousands of algorithms that we actually trained on the data. And we only use 60% of the data set available for training. We then use the next 20% for optimizing the model. And eventually we opened the last test sets that had been closed all through the modeling and the training of the algorithms. We opened that after selecting on the final model. We had specific criteria that we wanted to develop a robust model that would be robust in terms of missingness of specific types of data, meaning that if we took out a whole class of data, it could be pathology results, or it could be information about infections in a specific time interval, the model would still work. So we wanted to make sure that we had a robust model that would work across different health systems and different cohorts of patients. Next, obviously, we wanted to provide personalized risk factors, and we wanted to be able to model interactions between the different variables or features. And obviously, we wanted to account for nonlinearity when modeling. And here is just demonstrating the leukocyte count and the correlation with outcome and the correlation with the prediction. Just to emphasizing again that the same variable here, leukocyte count, could have a different impact on the prediction of the algorithm, obviously depending on the level of that variable, but also on the other variables available for that patient. And obviously also that the true outcome as demonstrated here would vary. So we need to allow for these nonlinear correlations, and we need to allow for the interactions. Eventually, we came up with a model based on 28 algorithms, genetic algorithms, including approximately 80 variables in terms of 228 features. And what you see here summarized is that the different algorithms all align in the bottom here. They select different variables that are listed on the right side here to be included for the modeling. And some of them only use one type of variables, that's the different colors included here, but most of them would actually use a number of different variables and different types of variables to predict the outcome. And then obviously we sum up the prediction and give an estimate for the individual patient. In this work, we also try to develop a doctor's choice model, because I may admit that I actually was of the belief that if we just choose the variables that we wanted to include based on our prior medical and laboratory knowledge, we would be able to design a much better model, because we would focus the algorithms on what we believe would be of importance to predict the outcome here, infections or need of CLL treatment. And we failed big time. So the data-driven approach, starting out with all the 1800 variables available, was much, much better than the doctor's choice model. And to my surprise, immunoglobulin levels that have been known for ages to be correlated with risk of infections, they were actually not included for the 80 top variables, not because they don't have a correlation, they have when we tested in the data set, but other variables had much more information to provide to the model. Next step after developing the model is actually to convince my fellow clinicians and our patients that it makes sense to use such a data-driven model to identify patients at increased risk here of infection to CLL treatment. Thus, we included explainability in terms of shared values for individual patients, and obviously also assessed across the population. But I guess most importantly, to be able to explain to the individual physician and the individual patient that this patient had a specific high risk due to these factors. And as you see in the bottom part here, IGHV unmutated status makes sense because we know it has a major impact on the outcome for the CLL treatment, but other variables like transferrin and number of SMOT cells, I had absolutely no idea that those would be part of predicting or impacting the risk of infections for patients with CLL. On the other hand, I had no clue that glucose levels would actually be also of importance here in terms of driving this patient a little more towards the low risk phenotype. In addition to that, we wanted to include a certainty estimate with the prediction to make sure that we would not jeopardize patients by putting them on a specific treatment or choosing a specific clinical trial or specific treatment and clinical practice for this patient. Thus, what you see here is that we allowed only patients in the yellow line on the upper left part here, which would be predicted as high risk and high confidence to be entered into a clinical trial that we developed. These patients had more than a 70% risk of developing a serious infection or receiving CLL treatment within 2 years from date of diagnosis. If we followed them for 5 years, it would be more than 95% of these patients experiencing either a serious infection or CLL treatment. And on the other hand, the blue graph up here is the predicted low risk patients with a very low occurrence of these events. To the upper right corner, you see that the first event with an infection or treatment was more or less equally divided across the population to the right with high risk. But to enter this algorithm or to introduce it and implement it into clinical use, we needed to prove that we could actually use it to improve outcome for our patients. Thus, we next designed a clinical trial, the Prevent the Call trial, where we only would include patients predicted as high risk, high confidence for 1-to-1 randomization between a very short 3-month treatment for the CLL disease. A short treatment period because we didn't want to put the patients at risk in terms of adverse events. As I demonstrated before, upon regular treatment, patients have a high risk of infections. But with a very brief 3-month period of treatment with 2 targeted agents that we knew would have synergy in terms of fighting the CLL, but also had indications that they actually could improve the immune function in CLL. And providing 3 cycles of treatment, not aiming for cure, but aiming for turning back the natural history of immune dysfunction in CLL towards a pre-malignant state, more or less resembling monoclonal B lymphocytosis, but aiming at having a less severe immune dysfunction for these patients. And obviously we had to prove that this worked, so we went for the 1-to-1 randomization in what we call the Prevent a Call trial, or the preemptive venetoclax acalabrutinib trial for CLL. Hopefully to improve the outcome for our patients. We are currently enrolling patients for this trial, as I don't know the outcome. But what I know is that we don't want to stop here. We want to continue what I consider the life cycle of medical artificial intelligence. In the upper left corner here, the clinical situation, identifying what we want to model, identifying the data at hand, predicting, developing the artificial intelligence algorithm, setting up how we want to test this in clinical practice. What you see here is the clinical trial. Imagine that we, in the future, hopefully, are going to develop thousands of new algorithms that will actually help the physicians with what we consider decision support in the clinical setting. It will probably not be possible to test all of those in clinical trials. Thus, together with Miaela and other collaborators, been discussing, can we actually develop a way of avoiding the clinical trial, but still making sure that we actually test rigorously whether new algorithms actually improve the outcome for our patients. And this is something that I really want to work on. How can we make sure that we can implement new algorithms, like imagine in a laboratory when you introduce a new laboratory test or new management of platelets for platelet transfusions? You would usually just make sure based on pre-testing that it seems to be safe, and then you would follow for, say, the next half year, how the outcome would be, how would it match prior testing, how would the platelet transfusion match what you used to see in terms of efficacy. So we might be able to develop, based on these huge amounts of data that we have available, a more direct way of implementing new algorithms into medical practice without jeopardizing the health of our patients. So we might find a way to speed up this surveillance of the life cycle of clinical algorithms. And obviously we need to make sure that we continue to develop the translational studies that will again feed back into the clinical training of new algorithms and improved algorithms by allowing more variables to be entered. And in terms of more variables, currently, as we look at immune dysfunction, we are working on characterizing the microbiome in CLL. So again, just to emphasizing that we don't need to stop with just working on one type of omics. We want to include a variety of different immune phenotyping approaches and different omics approaches to improve the prediction for our patients and to provide better decision support for physicians treating our patients. Thus, for clinical trials, we obviously need to address both the microbiome, what you see up here in the upper part, and the feces and saliva samples, but we also need to characterize from functional assays the blood and bone marrow and lymph nodes. So it's really in the design of clinical trial where we need, and in the implementation in clinical practice, where we need to make sure that we can actually collect these data that we're going to model. And sometimes when discussing with data scientists, it's discussing, we just want the data. We just want to be able to model them. We want the clean data. And just want to emphasize to you how much work it takes for our patients, for our support staff, for our students and our organization to actually assemble these data, to make sure they have a sufficient valid representative biobank with the different types of material that we want to use to model. So remember that for all of us, we're actually working on this, but it takes a lot of work to collect these kind of data. This means that we have a duty to use these data in collaborations across different European and worldwide study centers. What I represent here is the CLL Clue, an EU-funded project where we combine the clinical data as demonstrated here for CLL-TIM, together with the functional characterization of biobank samples, both from clinical trials and from regular biobanks from patients treated outside clinical trials. And we use this to develop new algorithms that represent new biomarkers or complex biomarkers to be used in novel clinical trials. So we want to make sure that we continue this life cycle. And I started out with presenting also genetic data. We only included for the CLL-TIM genetic data that would be analyzed in routine. That was the IGHV status and the FISH cytogenetic analysis. But obviously we wanted to test whether we could improve the prediction in CLL-TIM by adding on top of CLL-TIM the data on genetic mutations, driver mutations across patients with CLL. And this is work by Mehdi Parviz, who I believe is also present here today. So essentially, as demonstrated also for CLL-TIM, a prediction window, a 2-year and a 5-year outlook, the prediction point defined as 3 months after diagnosis of CLL, making sure that we could include different types of variables, different types of information to be presented to the model. We started out with what's called CLL-IPI, the gold standard of a prognostic model or prognostic marker, including 5 variables in CLL. Then we added subsequently the baseline characteristics, more or less what was represented in CLL-TIM, and the genetic information about mutations, the green and the red dots in the middle here. And we modeled the risk of death, the risk of receiving treatment, the risk of infection, and the composite outcome of an infection or CLL treatment that's also used in CLL-TIM. And what you see here is the representation of which variables would turn into most important for the different predictions. And I don't want you to go into details with this, but we could actually detect patterns that would indicate that early risk of infection within the first 2-year period might contain the same variables that were important for late fatalities within the 5-year outlook, indicating again, as I demonstrated all the way to the start, that patients with CLL actually die from infections. Thus again, being able to introduce explainability into the modeling here. And as you see here in the composite endpoint all the way to the right, that you have different variables impacting the risk of the composite endpoint, the part here in yellow, than what you have for treatment and infection independently, demonstrating again that modeling a composite outcome might improve the predictive performance of your algorithm. And what was most surprising to us, I believe, that in the upper left corner, if you only used the IPI factors here. It was not a very good model. It's only 5 variables included. If you go to the lower left part, we added in the genetic information about mutations, and it didn't improve at all, at least very little. On the other hand, in the upper right corner, if we added all the information from the electronic health records, it improved a lot. And in the lower right corner, combining them all, it actually didn't make much difference, but maybe a little improvement to add the driver mutations information about genetic events on top of the electronic health record data. There's been other studies more or less resembling this, illustrating that sometimes focusing on the genetics and the omics data in the lower left corner here kind of blind us from seeing that we already have a ton of data available for our patients that, as in this case, may improve the predictive performance much more than just looking at the genetic events. And it might seem a little boring to look for data in electronic health records, but it's just until you start looking at the amount of data. Just in eastern part of Denmark, representing, as you see here, 2.9 million citizens, we have an electronic health record system, the EPIC system. I think it's more or less the same you have in Cambridge, unless you got rid of it again. More than 50,000 end users, more than 15,000 daily users, a lot of workstations and medical equipment linked to it. And as you see in the bottom part here, more than 300 billion rows of data, more than 11,000 tables, and more than 300 of these tables with more than 50 million rows of data. So the amount of data available from our electronic health records, if we actually start to use them, provides a very, very rich source that we should never overlook when we try to develop new predictive algorithms, and that we should never overlook when we aim to create new omics data. We should always take into account that it might be that we already had these data available from the regular routine data analyzed. And I just want to emphasize to you again, when you start to come up with the proposals for new data analysis that we should definitely develop together, that it takes a lot of work to get these data in real time. What I've shown you here has been based on registry data, meaning that they have a delay approximately 1.5 years from time the patient has had an event until we can actually collect the data, because it has to go through several registries. A lot of it is actually manually entered. In the electronic health record system, we are not allowed to work in the Chronicles database, that's the real-time database, but we are able to get research extracts from the Caboodle database that has a 24-hour delay. If we had used data directly from the Caboodle, they were not present, or it was not possible for us when we developed the algorithm. If we had done that, we would actually be able to map directly back into the electronic health record system, the algorithm, and run it and give the prediction directly in the electronic health record system. This was not possible, thus we have spent the last half year to remap all the variables included for the CLL-TIM back into the Caboodle database to be able to provide the prediction here to my colleagues, demonstrating for the first time that we can actually get the prediction represented directly into the EPIC system, just with a 24-hour delay, meaning that rather than having to enter manually all these variables, we are able to provide to the treating physician the prediction just 24-hour delayed. We are still not able to do it in real time, but almost. And we want to go further. We want to use these type of different data types to train algorithms that can be developed and work across different data sets to allow for what we consider meta learning, meaning that we can bring the different data sets that we have across different medical conditions to life, And I just want to finish off with demonstrating that other diseases here, COVID-19 across all patients diagnosed with a positive test of COVID-19 in eastern part of Denmark, that they resemble some of the risk factors that we actually see in CLL. And by analyzing immune function here by a very standardized stimulated assay called True Culture, developed from the Institut Pasteur, we can inform our patients and our clinicians about immune dysfunction, where we learn from one disease here, COVID-19, and apply it afterwards in CLL. So I want to emphasize that we should make sure that we can reuse these data. And here again, the other way around, using the modeling approach in CLL here to model also based on the same type of data, but actually here access directly from the health record to model the risk for patients with testing positive with COVID-19 from time point of positive test to be able to allocate them to different treatment approaches. So summarizing up, our vision is to create a tool where we can actually make personalized treatment recommendations, make sure that we can enter the different modules, input modules here to the left, and by creating smart algorithms together with you, with the machine learning community, to be able to predict different outcomes, whether we want to predict the outcome on a specific treatment or the risk of a specific adverse event. So we need these safe data platforms for storage and modeling of all types of data, medical data here across different cohorts. We won't need to be able to segregate the different data types and be able to join them during modeling. And we need to be able to add molecular and genetic data from biobank samples, but we need to be able to add them on top of the data already available in the electronic health records. And we need a one-stop shop for approval of such projects and a path for data assembly and data sharing across different research communities. Because we need, and it's a duty for us as researchers, to bring these big registry and molecular data to live for our patients, to define together outcome definitions and develop features to make sure that we can actually develop what I would eventually consider artificial intelligence for medicine, and to test it in clinical trials, or to develop the next level of ongoing testing and validation of new algorithms implemented into clinical practice. Thank you so much for listening in. I'm looking very much forward to discuss this presentation, and obviously my thanks go to both the patients providing the data for this and my research group and my collaborators and the funding agencies. Thank you so much.
Thank you, Carsten, for a fantastic talk. Really, lots of food for thought for both, I think, the machine learning community and the clinical community. And it's wonderful to see how you have walked this walk of really using machine learning and making it useful for you. So it's wonderful to see that become a reality. I really hope that our center will be able to follow in the same footsteps as you did. I have many questions to you, but I think it's better to let others ask the questions, since I have the privilege to be able to ask the questions later. Any questions? So Tara, would you like to unmute yourself and maybe ask the question?
Yes, thank you very much. It was great. I really enjoyed your talk. Great stuff. So my question is, so I was thinking, for example, if you're a medical doctor and based on your expertise, you might be able to suggest a treatment option to these, people who have been predicted to have high risk of infections. How does it work from, medical ethics point of view? It is difficult from in silico modeling to jump into clinical interventions. You see what I mean? What is your opinion on that?
So it's a really important question, and thank you so much for bringing it up. That if we just make this information available to the clinician, we would kind of leave the clinician without any input on what to do, how to act on this. It could be like the easy way to implement a new algorithm in terms of what we would consider decision support. This is essentially what we're trying to do now with implementing it directly into the EPIC system, making it available to clinicians, the information. But we do it in a structured way where we only allow the clinicians to use it for selection of patients for clinical trial. Because exactly as you say, would it be ethically right to introduce a new treatment without monitoring whether it actually improves the outcome for the patients? So what we can do with the algorithm is to detect or identify the patients at high risk of a specific outcome. But at the moment, and I'm sure Mihaela and a lot of you would argue that we are getting very close to providing also information about what we should do for these patients. But still, we are left with the clinical randomized trial to assess what would actually improve the outcome for our patients. And I've been in thorough discussions with Mihaela, whether the work of Mihaela's group on synthetic twins and synthetic virtual twins and also other methods would allow us to use the already existing clinical data to provide and implement into clinical practice, kind of bypassing the clinical trial, new treatments or algorithms. But I'm not sure we are ready for main time yet for this approach.
Thank you
Thank
very
you,
much.
Carsten. Any other questions by anyone? We have time for a few more questions. Would you like to unmute yourself?
Yeah, thanks. Sorry, I was typing it away, but great talk, Carsten. Thank you very much. Resonates a lot with stuff we're trying to do here. I had a couple of questions, but perhaps first of all, you suggested that we could expand and improve on prediction by aligning genomic analyses, bringing in microbiome, bringing in other technologies that will measure more than what is routinely collected. But that's a bit of a problem because you also very nicely show that we don't know what to measure. So in other words, or we don't know what to use to predict. So amongst the things that clinicians already measure routinely, there are predictive features out there. We just don't know which ones they are and how to put them together, which is what your model has nicely done. But from a genomic perspective, if we start making many thousands of measurements on a comparatively small number of samples, that changes the problem into a different one of feature selection, where you've got far more features, far fewer samples, and how do we identify from that what features we should be adding into our models? And I'm just wondering if you've started to address that problem.
So thank you so much. It's really an important issue, and I think it comes down to numbers, because if we had unlimited access to patient data, we should not limit ourselves. We should actually make a fully data-driven selection of which variables would include information or hold information to predict or model which outcomes. And I'm maybe happy, maybe not. It depends on how you succeed to see that you are having the EPIC system implemented in a lot of UK hospitals and soon to be implemented in a number of others, because it holds a very rich data source. But it also holds a lot of problems in terms of accessing these data. We got EPIC introduced in 2016, and not until during the COVID-19 pandemic did we succeed as the first to actually get these data out from the system. So you really need to work on this. My suggestion would be to model the different types of data in what I would consider separate data lakes, and then join them to allow to use the information generated across different patient cohorts with each type of data to increase the understanding or the pattern recognition within these types of data, and then joining the different approaches to pattern recognition together to allow you actually to test also in smaller cohorts by reducing these the principal components to test whether you could actually improve the prediction. But eventually this come, and that's definitely outside my area of expertise, back to whether we can apply one-shot modeling in terms of learning the algorithms to learn from a number of small cohorts and making sure that we can model and account for both domain shifts in terms of types of data, but also domain shift in terms of type of disease that we want to model. So I don't have any easy answer to this. I believe that data sharing and managing the data privacy issues to actually share data across different research environments and allow for more open discussions and more open competitions on the same data would allow us to develop these approaches. But sorry for no easy answers to this.
Thanks, Carsten. I wasn't expecting an easy answer, but it's a good one. Thank you.
Wonderful. Any other questions?
Can I ask another one? Sorry to
Go ahead.
To do one more. I mean, you've described a lovely model development, and the route to actual implementation is a long and stony one. It's a difficult path, one that often takes you away from traditional academic routes where you don't get any credit until you've actually translated something fully into practice, which is extremely high risk. I mean, this space is changing in terms of the amount of regulation that's being brought in as well. So are you trying to implement this as a means of changing the way we treat CLL? And if so, how?
So in parallel, I'm also developing clinical trials with a number of my colleagues across the world, regular clinical trials. And I'm getting so frustrated by seeing that we, as part of these clinical trials, develop translational programs, develop more data, collect data. And after publishing the clinical trials, the data are more or less dead. We don't use them to develop the next step in terms of improving the outcome for our patients, identifying different subgroups of patients with specific treatment needs, and in the next clinical trial, test whether we can actually improve the outcome. So that's really what I'm aiming for by the Prevent a Call trial. But I may also admit that I met a lot of troubles I didn't expect. One of them being that we need still to enter the data de novo manually to make the prediction, because we are still struggling to validate that the prediction we develop within the EPIC system is exactly matching the prediction on the research data outside the EPIC system, because we had to remap everything. And I'm struggling to convince my colleagues, and I think that goes back to the question from Marta Milo. I have to convince my clinician, my clinical fellows, and my patients that it's actually safe to trust the algorithm. We included explainability, meaning that we could actually pinpoint the individual risk factors, and we included a certainty estimates with the prediction to say you actually have this very high risk and you're in the group where we had a certain prediction. And still close to half of the patients that have been identified as high risk, high confidence predictions refused to go on the clinical trial because they didn't want an algorithm to predict for them whether to receive treatment or not. And they didn't want to go into a randomized trial between nothing or just observation, which would be standard of care, and treatment based on an algorithm. So it's a major step. How do we actually introduce this into clinical use? And I believe for the first instances, we need clinical trials to actually prove that it's safe. So we're also following all the patients that are predicted as low confidence or low risk. And we are following the patients refusing to go on the trial. They still allow us to follow them and the outcome. So it's really a question about how to introduce this in a stepwise way. And on the other hand, I'm a very impatient researcher and clinician, so I want this to be done. I want us together to model these data and use these data because I believe it's a duty to our patients.
Brilliant. Thank you so much, Carsten, for this visionary work and important work. Thank you so much for visiting us virtually. We hope that you are going to visit us in person at some moment in time in the future as well. And I'm sure there'll be many, many more questions. So thank you very much.
And
thank
please
you,
feel
everyone.
free to reach out. I'm very happy to engage in further discussions offline as well. And thank you so much for listening in today.
Thank you.